Welcome to Biotanemy.
Bacterial transformation. Bacterial transformation is the process by which the bacteria absorb single-stranded DNA from their surrounding area. The transformation can be induced in the laboratory using calcium chloride precipitation or through electroporation. The process of transformation in bacteria was first demonstrated by British bacteriologist Frederick Griffith. In his experiment, Griffith takes two strains of Streptococcus pneumoniae bacterium, the non-pathogenic rough strain, which is not surrounded by a capsule, hence appeared as rough colonies, and pathogenic smooth strain. They have a capsule and appear as smooth colonies in the nutrient agar plate. Griffith injected living rough strain into the mice, and the mice lived. Hence, he injected the smooth strain of Streptococcus pneumoniae into another mouse. Within a few days, the mice died due to pneumonia. So, in another experiment, Griffith injected a heat-killed smooth strain of bacteria into mice. He found out that the mice lived. Hence, in the third set of experiments, Griffith injected a mixture of heat-killed smooth strain and live rough strain of bacteria into mouse, hoping it to live. But, to his surprise, the mouse died within a few days due to pneumonia. And Griffith has been able to recover live, smooth strains of bacteria from these dead mice. Griffith concluded that something from the heat-killed smooth strain of bacteria has been transferred into the live rough strain of bacteria, transforming the rough strain to smooth strain. He called this process as transforming principle. The transformation in modern biotechnology has many applications. For example, in molecular cloning, it will help in recombinant protein production, gene therapy, production of transgenic organisms, etc. Since it is important in modern biotechnology, how can we do it in a laboratory? The transformation in the laboratory is done in two steps. 1. Competent cell preparation. 2. Transformation. The competent cell preparation is the process of converting bacterial cells ready to take any DNA present in their surrounding. Not every bacterial cell is competent to take up the foreign DNA from the outside, and these bacterial cells are called uncompetent bacterial cells. The steps for converting uncompetent cells to competent cells are: 1. Preparation of 100 mg per ml of ampicillin stock. Add 300 microliter of ampicillin into 500 milliliter of Luria batoni agar or LB agar. 2. Take 1.5 milliliters of 3-hour incubated bacterial cells that are required to be transformed in an Eppendorf tube, and incubate them on ice for 5 minutes. 3. Centrifuge the cells at 4000 rpm for 4 minutes. Collect the pelleted cells. 4. Add 600 microliter of ice-chilled 0.1 molar calcium chloride to the pelleted cells, and incubate them in ice for 30 minutes. 5. Centrifuge them again at 4000 rpm for 4 minutes, and collect the pelleted cells. Now all the cells in the tube have become competent. The next step is to add DNA into the competent cells. For that, add 100 microliter of ice-chilled 0.1 molar calcium chloride, along with 5 microliters of DNA sample. Incubate it on ice for 30 minutes. To induce the cells to take up the DNA, give it a heat shock by keeping them at 42 degrees Celsius for 40 seconds. Immediately transfer the heat-shocked cells into ice, and keep them for 5 minutes. 6. Add 600 microliter LB broth into the transformed cells and incubate them at 37 degrees Celsius for 1 hour. 7. Plate 200 microliter of transformed cells to the ampicillin-containing plate. Incubate for 24 hours. Only transformed cells will grow in the medium containing ampicillin, since the original cells are sensitive to ampicillin, but the plasmid DNA has an ampicillin-resistant gene.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.